Okay, so my project is on the impact of anatomical subject specificity on the muscle moment arms in the cervical spine. So firstly, for some background, the human cervical spine is a complex biomechanical system, which is crucial for head movement and spinal cord protection, but it's highly susceptible to pathology such as neck pain, which is one of the most frequent musculoskeletal complaints. So therefore, understanding its biomechanics is essential for effective prevention and treatment strategies. In silico musculoskeletal modeling, particularly using platforms such as OpenSim, offers a cost-effective alternative to in vivo measurements for investigating cervical spine function. However, widely used generic musculoskeletal models are typically based on population average anthropometrics and often neglect significant intersubject anatomical variations in structures like joint centers and muscle attachment locations. While subject-specific models have been developed through anthropometric scaling, they often overlook the detailed impact of these anatomical variations on biomechanical predictions. Specifically, a direct comparison quantifying the effects of subject-specific anatomical parameters on the muscle moment arms of the cervical spine relative to generic scaling has been neglected currently. So for the aims and objectives of this project, the project aimed to develop a workflow to produce subject-specific musculoskeletal models of the cervical spine using CT image data and compare the muscle moment arms of these models to those of generic scale models through various spinal motions. This methodology could be applied to enable population-level computational modeling that may aid in the identification, prevention, and management of conditions related to the cervical spine. To achieve this, the following objectives were identified. Develop detailed segmentations of the cervical vertebrae and relevant muscles from CT image data for a range of healthy subjects. Secondly, generate surfaces from these segmentations and arrange these to develop subject-specific musculoskeletal models of the cervical spine. Then scale generic musculoskeletal models of the cervical spine using morphological parameters from the data sets to facilitate comparison with subject-specific models. And then finally, analyze and compare the moment arms of the selected muscles in the generic and subject-specific models through the full range of motion of the cervical spine. So, for the first part of the methodology, 22 CT images and segmentations of the cervical spine were obtained from the RSNA cervical spine fractures CT dataset. In these data, the individual vertebrae had been automatically segmented using a 3D UNet model. However, many of these, as you can see on the picture on the left, were of poor quality, where the vertebrae had been misidentified. Therefore, each slice was manually revised using ITK Snap software, as you can see on the right, where it's been revised. Additionally, segmentations of the muscles attaching to the cervical spine were produced using the automated total segmentator tool, which you can see on the right of the segmented muscles. Second part of the method was to develop surfaces and find joint center locations for the subjects. So the 3D surface models were then generated from the models using 3D slicer software. Individual transformation matrices were used to transform each surface from the global CT scan coordinate system to the surface's local coordinate system. An OpenSim cervical spine model previously developed was used as a base to determine the subject-specific joint center of rotations. The mid-sagittal cross-sectional areas of the vertebrae used in this model were measured, and the location of each joint center of rotation in the local coordinate system of their respective parent and child bodies were extracted from the OpenSim file. These center of rotation coordinates were transformed into the local coordinate systems of the vertebrae from the RSNA data and transformed into OpenSim's coordinate system. Finally, normalized center of rotation coordinates were calculated using the equation shown. As no information about the anthropometrics of each subject was provided in the dataset, a scaling approach using the cervical length of the assembled OpenSim models was applied using the Euclidean distance between the C1 and C7 vertebrae in each model. The next part of the method was to find subject-specific muscle attachment locations. So to do this, two axial planes were defined at the global Y coordinates of the muscle origin and insertion points indicated in in the base model. The global intersection points between these two planes and the muscle line of action were calculated, and each intersection point was then transformed into the local coordinate system of the original attachment body. So in image A here, you can see the original muscle attachment points using a generic model, and the revised, and image B shows the revised ones that were specific to each subject. Next was developing the models for testing. So a previous developed high-order models uses the base for all scaled and subject-specific models, and then 3 models were generated for each subject. So firstly, a generic scaled model, which was just this generic high-order model, scaled to the anthropometry of each subject using the length of the cervical spine as a scaling parameter. Secondly was a hybrid model, which replaced the vertebral body geometries with subject-specific ones from the CT data, and as well as the CT data joint center of rotation locations, but they maintained the relative muscle attachment locations used in the high-order model. And the rest of the bodies in the model were scaled in the same way as the generic scaled model. And then finally, the subject-specific model was the same as the hybrid model, but it also had subject-specific muscle attachment locations. Now, the next part was preparing for the muscle moment and arm analysis. So firstly, motion files simulating the rotation of the joints through the full range of motion were required and generated. So this was for flexion extension, lateral bending, and axial rotation. So on the image on the left here, you can see the full range of axial rotation, and on the right is the full range of flexion extension used. And then following this, the muscle moment arms in each OpenSim model were computed through the coordinate range of each motion file using the Analyze tool in the OpenSim. The moment arms were determined for all muscles using each joint in the model in turn as a reference joint. Statistical differences between the 3 model types were determined using statistical parametric mapping. Additionally, asymmetries in left and right muscle moment arm curves for the subject-specific models were assessed for. And the next step was to evaluate the accuracy of the model. So the position of the joint center rotations was evaluated by forcing the subject-specific OpenSim models into the pose of the subjects when the CT scan was taken, and then comparing the positional differences between adjacent vertebrae for the CT data and the OpenSim models. And you can see the results of this on the left here. So the main takeaways from this was that the greatest difference in joint center rotation location between the OpenSim model and CT data was in the Y component of the C1 to C2 joint level, with a mean positional error of minus 11.8 millimeters. And then I conclude that this error was likely because of the likely inherited from the base high-order model, an error in that. And then to assess the accuracy of muscle attachment locations, I plotted the mean positions of each vertebral centroid relative to C7, which is shown on the right here. So this was important because subject-specific muscle attachment locations were estimated based on the line of action of muscles segmented from the CT scan. However, this estimation operated on the assumption that the lines of action of the muscles in the CT scan were similar to the lines of action in the neutral spine alignment used in the OpenSim model. As a result, deviation from the spine alignment in the CT pose from the OpenSim alignment would likely reduce the accuracy of muscle attachment locations. So any variation that you can see here on the right likely reduced the accuracy of muscle attachment locations. So finally, the results. So firstly was the variability between the subject-specific models and the generic scale models, which was the main aim of this project. So the subject-specific models presented the widest distribution of moment arm lengths for all investigated muscles over all three motions, followed by the hybrid models and the generic scale models. To assess for left and right side symmetry, the magnitude of muscle moment arms were compared in flexion extension, and the muscle moment arms of the agonist muscles over their magnitude of lateral bending and axial rotations were also compared. No significant differences were found between left side and right side muscle moment arms in flexion extension. However, significant differences were reported between left and right scalenus anterior and scalenus medius in lateral bending, and between the left and right levator scapula and scalenus anterior in axial rotation. So the main discussion points of this. So the project successfully developed a CT-based workflow for generating subject-specific musculoskeletal models of the cervical spine and provided the first comprehensive comparison of their predicted muscle moment arms against generic scale models. The findings underscore that simple anthropometric scaling using generic models fails to adequately capture the anatomical variability within populations, leading to reduced accuracy in muscle moment arm predictions. The increase inter-subject muscle moment arm variation observed in subject-specific models highlights the critical importance of incorporating individual anatomic detail. The significant differences in muscle moment arms between model types confirm that subject-specific anatomical parameters substantially alter biomechanical predictions. While some differences were consistent offsets, others varied with joint rotation angle, which indicates a complex interaction between joint axes and muscle lines of action. Instances where no significant differences were found suggests that generic scaling might sometimes approximate mean muscle moment arms, but crucially misses the true intersubject variability. The observed left-right asymmetries in lateral bending and axial rotation muscle moment arms are likely artifacts of the subject's supine CT scan poses rather than true physiological differences between the left and right side muscles. Despite this, there were some limitations to this study, which primarily stemmed from the source CT data due to the limited field of view in the CT scans, which required the extrapolation of muscle lines of action to estimate their attachment points, and the CT scan differing from the OpenSim neutral pose. And then secondly, there were limitations in methodological aspects relating to the time-consuming nature of manual segmentation and the reliance on the reference model, which may have been flawed for the joint center rotation estimation. And then future work on this should focus on refining joint center rotation determination to better match CT data, and then also improving muscle attachment estimation, exploring nonlinear muscle paths, and critically validating these model predictions against in vivo data using methods such as the tendon excursion or EMG measurements to confirm the accuracy of these predictions. So the key conclusions are that the inclusion of subject-specific joint centers of rotation and muscle attachments increase intersubject variation in predicted muscle moment arms compared to generic scaled models. This highlights that simple anthropometric scaling poorly captures differences in moment arms resulting from anatomical variability. Significant differences in muscle moment arm profiles were identified across various muscles and motions when comparing generic scaled to subject-specific models. Some comparisons exhibited relatively consistent differences with low coefficients of variation, which suggests geometric shifts in joint center rotation or muscle attachment locations, while other exhibited differences highly dependent on the joint rotation angle, indicating that there were different joint axes of muscle lines of action between these model types. So in conclusion, the developed workflow for subject-specific musculoskeletal models of the cervical spine using CT data has been successfully applied to 22 subjects. The comparison of muscle moment arms predicted for generic scaled and subject-specific models conducted for the first time demonstrates that incorporating anatomical subject specificity is essential for accurately modeling the moment arms in the human neck muscles. And then just as a summary of this project, I developed a CT-based workflow for subject-specific cervical spine models in paired moment arms, predicted these generic scale models for the first time. And then anatomical subject specificity significantly alters moment arm predictions and captures greater intersubject variability compared to generic scaling. However, there were some limited some limitations to this, and future work should focus on refining these methods to overcome these limitations and also validating these predictions against in vivo data. So thank you for listening to this presentation. Do you have any questions?
Thank you, Chris.
Thank you very much, Chris. Alexandra, I don't know if you want to go first, otherwise I have a couple of questions for Chris.
Ellen is here, and I would let her go first because imaging is her area.
Hi there. Great presentation, Chris. I really enjoyed it. A really nice piece of work. I missed the beginning of your presentation. I'm extremely sorry for that. There was a mix-up with my calendar. But I was really interested in... Is there an echo when I'm speaking?
It is. I think it might be coming from... I'm just going to knock off a few people's audio. Just hold on a sec. Okay, try that again, Helen.
Okay. So you mentioned, or you've described, a CT workflow. It certainly looks like this is the basis of something that could be very useful to other researchers and clinicians in the future. Are there plans for how this might be made accessible to other interested parties?
Not currently, but I was working with my supervisor and some postgrad students. I gave some of my code to them, so I think they're currently working on building on this project.
That's great. And I mean, to analyse 22 subjects is already quite a commendable undertaking. You talked about some of the ways that you might be able to refine and improve the algorithm moving forward. And one of the things that you mentioned was associated with the accurate determination of the muscle attachment and therefore how you apply the analysis. Did you look at the errors associated with the human aspect of determining the muscle segmentation from your CT images?
Yeah, so the muscle segmentations were determined using an automated method, but the problem with that is the low resolution of the CT images, and because the muscles are less dense, you can't see them as easily. So there are quite a few errors in their segmentations. So it was likely that they wouldn't be completely representative. And also, to simplify, I modeled them as I modeled the muscles as straight lines with a single attachment and insertion point, but which isn't always technically true. And there are other muscles with multiple attachment points that can't be modeled as straight lines. So I think this could be improved by looking into muscles with various attachment points.
Okay. So if you accept that that is a sort of bounded limitation of the approach because of the technical aspects you just described associated with resolution, is there a way of managing that, or is it simply that you have to have in vivo data, movement data?
I think because it was an automated method, it could be improved by doing it manually or having a wider range of views, because I didn't have the full view of the muscle, so I had to extrapolate their lines of action. But if I had CT data with a wider view, I wouldn't have had to use that method.
Sure. Okay. Alex, do you have any further questions?
First, I would like to say that this is excellent study design, very systematic approach. I really like it. I would like to ask about statistical analysis. I saw that you adopted P0025. Why that value?
I think it was just a standard value generally used. So, and there was.
Okay, right, because at least in my area it's 005 or 001, so you could be easier on yourself with 005. But you analyzed all this, you did not account for multiple comparison?
So I compared between the three model types through each range of through each motion file. So through the full range of motion for example, flexion extension, I compared the subject-specific model, generic model, and the hybrid model to identify any differences between the three models.
Yes, but just I want to say, when you have multiple comparisons, when you have lots of comparisons, then you apply some additional procedures to account for that, because let's say for 5% error of 100, you would have 5 of them that would be false positive. But it doesn't matter, it's just minor, and it's quite advanced, I think, for you've done a great job. So which method you use for statistics?
Statistical parametric mapping through ANOVA.
Okay, I know. Okay, good. So yeah, that's all for me. Thank you.
I mean, the last two questions I had for you, Chris, which apologies because I'm gonna go a bit like further back from the start of your presentation compared to what Helen and Alexander have asked you. You mentioned the RSNA data set, which I mean, I'm not very familiar with because this is quite removed from the field I used to be working in. Could you just explain what that data set is and how did you pick those 22 subjects from that data set?
It's a data set from a variety of sources containing a few thousand subjects, and then I think 89 of them had already had segmentations done from them. So I selected. I basically went through the 89 and selected spines that were healthy and rejected ones with fractures or misalignments. So from that, I came up with 44 healthy subjects and then selected 22 from that data set. Sorry, I think your mic is muted.
Sorry, I was just going to ask you, how did you get from, I mean, those like healthy patient, I think you said 44 down to 22, that's like 50% selection. How did you select them in the end?
Well, because of I wanted to use the my new segmentation method, and then that is quite a time-consuming process. So I had to basically just choose. I chose 22 basically because of time limitations with this project.
Sorry, Sam, I have to keep muting you because there's some kind of echo going on from your side. So, all right, no worries. Thank you. Chris, could I just ask a question actually, because you brought up a really interesting point. How do you think your study, having looked at a very specific set of, shall we say, good quality spines, how do you think that your work would translate into looking at the rest of that data set that you set aside in terms of damaged spines and things? And do you think that there is an opportunity perhaps in further work to look at how it might translate into that, because you're going to see obviously some big variations? What are your thoughts on that?
I think that would be interesting to see, definitely. It may require some different kind of segmentation method, because I think I used a mainly automated based segmentation method and then manually cleaned it up. But with some of these spines, I think the segmentation can completely fail because of the fractures in them. So it would take more work to do that. And I think there would certainly be a lot more variation in these specific subjects of muscle memory arms because they would not perform as you expect for the average person in the population.
So do you think then that looking at maybe developing the equations for predicting it would be something that you would want to do in the future, perhaps?
Yeah, definitely. I think that would be a great way to build on this work, and then having potentially having an increased data set through the wider population could further back up these conclusions. And then obviously having this sort of data for damaged spines, I think, would be potentially even more useful, because these are the people that are more likely to experience pathology. So having data on this could be useful for creating effective prevention and treatment strategies.
Great, thank you very much. Anything else from our questioners? I think that's all then. Sam, are you good?
I'm very good. Thank you very much. And thank you, Chris, for...
All right, excellent.
Thank you, Chris.
Thank you.
Thank you, Chris.
Thanks very much, Chris.
Very good afternoon. Thanks for giving this opportunity to present today. My name is Vikas, Huraliganganahalli, Balaramegowda. I completed Masters in Biomedical Science at Swansea University, and the research topic on investigation on effect of carbon blood on blood clotting assessed by rotational thermometry under the supervision of Dr. Carl Lankins. Today I'll be cover introduction, aim and objectives, methods, results, discussion and conclusion, followed by further direction. Let's move on to introduction. Carbon block nanoparticles are widely used in industrial products such as tires, inks, and plastics, biomedical applications, and pharmaceutical. Because of the widespread use of the millions of workers are exposed to mainly through inhalation in their workspace area. One of the epidemiological studies have shown that exposure to the workers have 20 to 40% having higher risk of cardiovascular disease, including heart attacks, strokes, and blood clotting, this type of disorders we can see. However, the biological mechanism of reason behind that, it's not clear and still showing to having the risk. That's why one of the possible explanation I took that carbon block may influence the blood clotting. Therefore, this study aims to investigate whether carbon block alters the coagulation pathways, or if it's alter what concentration, and if its concentrations also giving higher, in what doses. We are going to see in further in the studies. Let's move on to our aims and objectives. The aim of our study was investigated carbon block nanoparticle effects on blood clotting using ROTEM. I examined both intrinsic and extrinsic pathways, but tested at multiple doses. Our key objectives for 4 main key objectives of our project. I investigated the role of carbon block nanoparticle in the process of blood clot formation. Secondly, I assessed the impact of intrinsic pathway. Thirdly, I assessed the impact of extrinsic pathway. Fourthly, I determined dose relation across the dose relationship across the all carbon black concentration. Now let's move on to the methods. I done the in vitro study in my research topic. The blood samples are collected from 5 healthy donors, and I conducted this all through one of the ethical. I conducted getting the permission through ethical approval from Swansea Ethical Committee. Then a lot of concentration carbon block concentration dose are tested, and 0. We receive control without having any carbon black nanoparticle. And secondly, the low medium carbon block concentration had a 0.1 microliter. Thirdly, there was medium 1 microliter of carbon black nanoparticles had it. Fourthly, we tested concentration on that high dose like 10 microliter. We used to identify those concentrations ROTEM. ROTEM is one of the good equipment to analyze the real clot formation. In the ROTEM, we analyzed extern means extrinsic pathway, and secondly, we analyzed the intrinsic pathway. Thirdly, we measure other clotting parameters: clotting time, clot formation time, clot strength. This gives the overall importance of how clot formation happens. That's why we tested these things. Then let's move on to our result. The clotting time, clot formation time, alpha angle, MCF. These are the parameters I use to identify X time and in time. The X times are initially decreased because initially we look into X time. The clotting time was decreased because less than 0.05. Clot formation time also decreased because less than 0.5. But alpha angle and MCF no changes because there is p value is greater than 0.5. When you look at into in time, the clotting time and alpha clot formation time, alpha angle, MCF, there is no changes because all are above p greater than 0.05. The overall result summary is the carbon black is clot initiation means clotting initiation only it will showing the decreases clotting time clot form but not in clot structure like alpha angle MCF there is no changes we can see and these are the graphical representation I done my in results. This is one of the example I done 8 graphs in overall in time 4 X time 4. So this is one example I'm putting here. This is our concentration control 0.1 molar and mg 1 micromilliliter and 10 microliter. This is our time 0, 50, 100 like this. And major in this graph we can observe here dramatic decrease. Control is without adding carbon black 170 seconds clotting time took and low dose 0.1 microliter 60 seconds we can see here and the high dose is 110 seconds we can see the clotting time. The carbon black only isolate clot formation extensive pathway because it's showing the only decreasing time. It's showing the effect only lower doses. P value less than 0. This is intent. There is no changes. You can still look at the graph. This is our concentration. This is our clotting time. All are similar clotting time. There is no changes. That's why carbon black doesn't affect the intrinsic pathway. P value is greater than 0.05. Now let's move on to discussion. Why extrinsic pathway only affect? Because carbon black particle binds to the factors like factor 8, 9 in the clotting parameter. That's why when it's bind to the parameter, automatically coagulation cascade is happen. When coagulation cascade is happen, automatically shows the cardiovascular diseases. When carbon black, then carbon nanoparticles enters the blood. It's when it enters the blood, it automatically binds to the protein. Once proteins are binded, the protein corona formations are formed. That's why these factors are on particle surface. When, however, the intrinsic factors are never affected to clotting factor 8, 9, 10, don't bind any carbon radially. And why? Sorry. So why no alpha angle and clot strength are changes? For example, no alpha angles are measures only fibrin polymerization. Carbon black doesn't affect this. And secondly, MCF is also final cause strength. It's only measures MCF is clot strength and clot stability. Even carbon black doesn't alter this. The only effect we can see is clot initiation, not clot structure. We also analyze the non-linear dose relationship because we need to know the linearity of the study of our graph. Like the strongest effects are shown low dose, 0.1 microliter. It's not in higher doses. Because in higher doses, the particles are automatically aggregated and reduce the surface area. Major impact of our dissection is low level of work culture may pose to greater thrombotics than we assume. Then conclusion. Our study suggests that carbon block selectively activates the extrinsic pathway, providing mechanistic link to cardiovascular disease in exposed workers in their workspace area. Secondly, our study demonstrated strongest effect on lower concentration, but challenging the traditional toxicology assumption and current safety limits because we can't say even lesser number of limits also used to carbon block. Lesser number of microliter we can use in any medical equipment because even our study suggested even lesser number of also showing the effect. That's why we need to review the safety limits. Thirdly, our study suggested carbon block affects the clot initiation, but not clot structure. It means only initiation showing the effect, not overall clot stability showing the. This needs further study, further investigation. Let's go for further direction. Means I done only in vitro study. Further direction is in vivo for animal studies to validate the finding. Secondly, it's protein corona characterization. If you know the particularly what protein corona characterization particular test of carbon, we can know more. But in clinical studies, expose carbon dioxide exposed to workers and chronic exposure of assessment. If we do this all the things, we can see more how carbon blacks particularly affect the persons in the real world. Our study limitation, we took only in vitro and small number of people and healthy donors only. And these are the reference I used in my project. And thank you. Is there any questions?
Thanks, Vikas. That's really fascinating. I'm going to pass over to Sarah and Sam to ask you a few questions, I think.
Sure. Thank you very much.
Thank you, Vikas. Sarah, do you want to go first, or should I?
Yeah, so it was a really, really interesting presentation. Thank you very much. So I want to start kind of in your methods section.
Yep.
You looked at different concentrations of your carbon black. How did you decide on those relative to the population exposure that you were seeing?
Actually, there is no particular study worked on the concentration presently. That's why we decided, based on our professor guidance, what they said, we just took the 0.1 molar. We can see first in range of 0.1 and 1 and 10 microliter. High doses and low doses. If it works, if it's using very less, if it works to very less doses, we can suggest the real world, it will not affecting the world. But it's showing our study, even low doses also affect, even high doses also affect. We can't give guarantee. We check with both, even less and higher, but it's both are showing lower dose also showing effect. That's why the study gives to further investigation on regarding that. We selected basis on our in-time and X-time values and clot formation timing.
So yeah, so that follow-on study looking at that clinical environment will really aid this.
Exactly.
Brilliant. Thank you. Have you got a question, Sam?
Yeah, thank you. I have a couple questions for you, Vikas. I mean, it was like a really nice study, very elegant and quite never heard of carbon particles before, or the impact on cardiovascular health. That was a bit of a sorry. That was the timer for your presentation. So the two questions I had for you was, I mean, the first one, could you describe a bit a bit more the sort of like in silico set you've used? Did you use
Yes.
like, I don't know, like a
Exactly.
phantom or like an in silico vessel in which you inserted blood? I mean, would be quite curious yet to know a bit more about how you conducted those experiments.
Yep. Thanks for your question. The carbon black nanoparticles are used in biomedical equipments like, for example, pacemakers or, or in medical diagnostics for analyzing blood clotting. There is a lot of equipment in pharmaceutical, in sunscreens, or in generally they are using in exposed, identifying the particular blood clottings. 8 million tons are used in producing work, and they can get everywhere. Clinical implication side effects become present on all over the world, like in all over the places. We can't say the particular one place is affected to one or one like one medically application only affect those people. No, you can affect everywhere, but we suggest why we chosen with carbon black nanoparticle in general world, getting highest cardiovascular risk. Cardiovascular is a major concern of the present world, showing important. That's why we selected a carbon black nanoparticle, because nanomaterials are very using in nowadays too much, getting easily bind with the platelets in blood. That's why they are using in everywhere, medically, pacemaker, biomedical equipment, pharmaceuticals, or in tires, rings, generally any equipment. They are using to in medicines or tablets. That's why we took the real why they are using to, even though there is a lot of side effects, we can't. Our study presents that even though they they are using too much, until unless without knowing the side effects of getting cardiovascular risk, we give the one indication there's even low amount of using carbon black, also very wrong amount. Like that we are giving generally. Is that clear? Is there anything your questions?
Yeah, no, thank you. I think you answered the bulk of the question. I was the main point of the question I asked. I mean, pretty much at time, so I don't know, Helen, if there's time for like one last question. If not, we can leave it to here maybe.
Yeah, I'm curious to know actually. Sam, I'm going to turn your audio off again. Sorry. Because I'm curious to know. I mean, obviously, carbon black is something that we see both in industrial environments as well as from cars and other things. Have you considered looking at particulate size in terms of its effect, or is this something that you would think of looking to do in the future?
It's not like a size, it's overall. For example, carbon black, it's not only affecting it's not only giving, it's giving benefits also. It's not only effects. If we do higher or lesser concentration, or we can suggest in vivo concentration, we can get to a clear idea if it's good for further investigation. Because for example, I done 0.1, 0.1 microliter or 10 microliter. If you change the other concentration like 15 microliter or 0.6 microliter, any other concentration, we can get to know that it's what are the its effect showing or not. When we are doing different or more healthy volunteers, or we can take with like that, if we change all the everything or in vivo studies, we are one doing in vitro, right? Based on that, I can't give conclusion. If we do all the studies and all the like in vivo or changing L questionnaires and every objectives, only we are doing clotting time, clot formation time, and those things. We can also do prothrombin time, platelets time. If you check all those things further investigation, it gives better to give the world real, it's it effective or not. Can give then conclude the what happened, because research means we can't give conclude one point. We need to give further investigation one by one by one, like that.
Yeah.
That's
Thank you very much.
All right, thank you. So, hi everyone. My name is Ekgari Kasawala, and I'm a PhD student at Aston University. And my PhD project it mainly focuses on assessing and evaluating usability and acceptance of wearable health devices for monitoring, assisting, and protecting other adults. So today I'll be focusing more on how I'm using TinyML and position-independent recognition of activity of daily living for the other adult demographic. So as we know, in the UK, we're currently facing a demographic shift. So we have more older adults over the age of 65 than we do have children under the age of 5. And 1 in 4 of these older adults have 2 or more chronic conditions. So these older adults are living longer, but they're often in poor health. And it creates a rising demand for healthcare support and independence. And wearable devices and Internet of Medical Things devices present an opportunity to assist with self-health management and also remote monitoring in smart healthcare systems to help assist in chronic disease management and also in continuous monitoring of older adults. And this is just a few examples of the type of sensors and research and the diseases that research has shown that they can help assist with. But despite these potential benefits, older adults underutilize these technologies compared to other age groups. And the few that do use these devices, they stop using them after a while. So this technology that could help is not being used. So my project aims to investigate methods of improving the usability challenges that older adults face when using these devices. And in that way, we'll be able to improve acceptance and also maximizing the benefits that older adults can gain from using these devices. To kind of like build a background and also understand how to move forward from this problem, I conducted a systematic review to understand the barriers and facilitators of this and adoption. I conducted 3 surveys and also performed a co-design workshop. And on top of this, I volunteer as a digital champion at AGK, where I support other adults in learning how to use technology. So from this, I've been able to gather insights, but way more than this, but I'll be able to share only a few that pertain to the approach that I'm talking about today. So the first one, on the market and device landscape, there's a huge oversaturation of wearable devices. So most older adults struggle to choose devices that they should use because there's a huge knowledge gap in understanding what information is good for them and what isn't. And because most of these devices do the same things, they're very unlikely. So designing a new device is very unlikely to change their perception of these devices and also change their decision to adopt. Secondly, on comfort and ergonomics. So they've expressed discomfort with the straps and also that they're restricted to wear it on the wrist. And some of them mentioned that they struggle with wearing the devices in warm weather because they sweat a lot and also during sleep. There's issues with discretion and identity, and most of them mentioned that using these kind of devices, specifically when applied for detection, they don't want to appear vulnerable to their family and their friends, so they'd rather not wear these devices at all. And some mentioned that these devices look so much like medical devices, so they don't want to wear them because it doesn't go with their clothing, and they would rather wear them normal, like with the classical watch face and things like that. And lastly, there's issues for on motivation and privacy. So there's very fluctuating daily motivation and also concerns about data privacy. And one main point from this was that because wearable devices, to see the benefits, you'd have to wear them for a long time. And most people get demotivated with wearing these devices if they're not getting immediate benefits. So that's a huge factor for this older adult demographic as well. So just a few definitions here. So activities of daily living when it comes to older adults is pretty much just tasks that are necessary for independent living. So this can include personal care and household activities. So from mobility to bathing to feeding, toileting, dressing, and transferring. And these activities are very important to monitor because they can help detect early indicators of cognitive decline, physical impairments, and also to help facilitate timely, integrated, and targeted interventions and just improve overall quality of life. So there's 2 different approaches of monitoring these activities of daily living. Some people use environmental sensors and smart home systems. These are very good because there's high compliance in that they're not intrusive and they require little noise interaction. And the bad side of this is that they're limited to monitored environments. So if people step out of the home, they cannot be monitored, and they can also be costly to install and maintain. There's also privacy concerns about systems that use cameras and audio for monitoring this type of activities. On the other hand, we have wearable systems which are portable and personal, and they can be worn outside the home. And the bad side of this is that they require regular charging and wearing, so you have to wear it all the time to be able to get the data from the wearable device. And as I mentioned earlier, there's usability challenges that come with this, and also the compliance and acceptance issue among the older adult demographic. So AI and machine learning have been used to improve measurement accuracy and also to optimize sensor placement in human activity recognition. But the gap is that most of the models that are used in this area, they're only looking at specified sensor placements. So most of them also use offline data sets. So they only evaluate offline. There's no actual real-world deployment. And most of them use research-grade sensors that provide the data quality, but they're very impractical for everyday users if this were to be implemented in the real world. So there's less scalability for this type of solutions. So in the study, we were looking at a proof of concept through time position independent recognition system using a consumer grade wearable device. And this would allow for flexible placement and therefore improve real world adoption by integrating with other adults' daily life. So for this study, only one participant was used, and we collected about 30 trials and 50 seconds per trial for all 4 activities. So the wearer was placed on 4 locations: on the shirt pocket, wrist, trouser pocket, and the ankle. And activities that were recorded were walking, stair descent, stair ascent, and idle state, where the user was just sitting down. And I used the platform Bus for this, where I uploaded the data and did my ML training and testing on there. And I used the raw data DSB block, which extracted about 36 features for this. And then from this, I used 3 dense layers with 100 epochs, and it was intentionally used with no location information from the collected data. And then from this, the model was then converted to a FLOT32 and then a BIS64 model conversion, and then deployed on the device to test real-world performance. From this, we're able to see that the average accuracy across all 4 locations, from the wrist, shirt pocket, trouser pocket, and the ankle, that the locations were above 80% for all locations, although the ankle position did have a lot of variance compared to like the shirt pocket. And this may be largely attributed to like the range of motion and the variations in movement when you're walking with something on your chest compared to the ankle, and especially with the variations in the activities that were recorded as well. But overall, training was done with about 92.8% accuracy and 90.59 accuracy for testing, and a slight drop for on-device inference as well. And during the testing phase, there was a convergence plateau at about 80 to 90%. So that might be the optimal epochs for the training the model. We also looked at the error rate. So this was mostly to see what activities are being misclassified as what activities. So from this, we're able to gather that stair ascent was underdetected, and stair descent was often misclassified as other movement. And this was asymmetrical, so between the stair ascent and stair descent. And this just suggests as well, like the accuracy for the overall accuracy of the system might perform more, but then activity-specific vulnerabilities are there as well. So stair-related actions are more prone to changes in kinematic features, and this might be something that will need to be looked into more. And the best performing activity was either state with less misclassifications. So from this, we're able to see that the model generalizes well in this data set for the training and testing, and that on-device inference is feasible on a consumer-grade wearable device. There was the issue with a small sample size, so this restricts the generalization of the study. And also there's very limited ADL types. So using 4 types may not translate very well into clinical practice. And also there was a lack of the order added participants. However, from the time I submitted for the competition, we've been able to implement a few changes that have then been accepted for a conference that is happening early March. And then from this, we've been able to compare time and frequency domain features, and also to use spectral analysis for the features. And then in combined sensor fusion with magnetometer and accelerometer data, I've been able to do my participant investigations, about 10 so far, and then currently working on expanding the model between classical machine learning models, neural networks, and also hybrid architectures, and then to see how they compare on deployability on this. And then end goal for this would then be to test on a target demographic and real life settings, and also to examine short-term and long-term user compliance, where ability to see if these changes would improve acceptance and usability in this demographic. Thank you.
Thank you very much. Right. I'm going to ask speakers or the questioners to ask some questions. Sam, if you want to be typing in your question, and we'll let other people ask questions first, if that makes it easier for you.
Okay, I'll start. So, yeah, very good presentation, a good demonstration of lots of knowledge of technical skills, technology. Is this part of your PhD?
Yes.
Okay. So, question. I understand that this is for individual use. So basically, the idea is that this device is trained for that particular person, right?
The for this particular study, it was just for one particular person, but so far we've collected data from multiple participants, and it's showing to generalize well between participants. So we tested 10 participants on we collected data from 10 participants and then tested the same model on 10 new participants, and we were still able to get accuracies about 80% from
Okay,
that.
this is what I wanted to ask because just one person, that's not much really, even for the. Okay, it's enough
Yeah.
to show proof of concept that you know how to use technology. It works, but
Yeah.
for validation of results, it's not much. But okay,
Yeah.
I want to ask you why these four particular activities?
Oh, the main thing that we did that for it was because some of the activities were very time consuming. So most people were not interested in doing the activities for a long period of time. So we kind of restricted to this four easy activities to do for a short while. But then we want to extend this to activities that other others can do themselves. For example, sit to stand or reaching and things like that. So like they can be actionable for clinical activities, I think.
That's what I wanted to ask. Like, sit to stand, something is that I would definitively expect, right? Ascending or descending, that's really challenging, I would say, to classify. So you give yourself hard times with that. I guess if you got a sit to stand, which is also clinically relevant. About your protocols, I want to ask you this feed-forward network. This is architecture, but what is the training protocol?
I didn't include it in the slides, but I used a platform called Agent Bus, which is used for Edge AI. So
This is
you can train
platform,
models on.
but what sort of is this feed for, or is this back propagation, or which is the like this the feed forward network? It's very wide. It's basically just showing that signal is not going backwards. And yeah, but what is the algorithm for training then? Is there any particular
On
name
the
of this algorithm?
No, because it's not a custom model that I developed. I use the Agent Bus platform that has these models already on the
Right.
platform,
Okay.
and you
Okay.
choose. Yeah, yeah,
And one more technical question also. You
yeah,
mentioned that, okay, you're using Edge Impulse platform and taking
yeah.
1,000 window data, basically 1 second, and applying digital signal processing, right? And but then you said that you're having this was done with 500 milliseconds window stride with zero padding. So why would you need zero padding on 500 milliseconds? You have like 15 seconds of data and once you were extracting 1 second. So I don't understand what were you doing in meanwhile. So you had 1 second, and then the next sentence you said you have half second and you were zero padding. Why would that be necessary?
Or it's just so the windows don't overlap for the selected windows. I'm not sure if I understand your question. Sorry.
Because okay, in your text you said each 1,000 millisecond window data was transformed into 36 feature input vector and so on. And then you said this was done with 500 millisecond window stride with zero padding.
Also, it does the stride for every 500 milliseconds, but the windows don't overlap.
Okay, right.
For the
Because it was that's a bit, yeah. Your results in figure 2, for which location is this?
figure 2 in the, I'll just open my document. Sorry.
These are like 4 groups and classification accuracy. And then
Yeah.
you said that, yeah, walking versus idle, stare versus walking, and so on. So, and you have 4 locations: wrist, shirt pocket, trouser pocket, and ankle. For which location is this?
All locations. So average accuracy for all locations.
So this is average.
Yeah. With the variance, I put the variance
Yeah.
of the accuracies.
Okay.
Yeah.
Okay. And I guess you perform some statistical analysis when you claim that there is no difference between locations.
Yeah, I did the kappa coefficient, I think. I'll just open my document, sorry. I have it with me.
Okay, but you did perform some statistical analysis on this.
Yeah.
Okay.
I did the Cohen's kappa statistic to just see if there was differences between the locations.
Okay.
And
Okay.
also, yeah.
So I wanted to ask you what's next, but you already replied and said that you collected more data on more participants. So that's all of me. Thank you.
All right,
Good luck
thank
with
you.
the study.
Thanks, Alessandra.
Thank you.
Samantha has got a couple of questions. She said, great presentation.
Thanks.
Are the activities likely to be used for rehabilitation process or for something else? Is her first question.
So what we're looking at is more like for people that live by themselves in their home, so you know if they're gotten up that day, if they've done anything in the home. So this would be the wearable is able to classify those data on device, and then because of data privacy issues and everything else, and also just in general, if we're sending all the data to cloud for processing, that would be energy consumption on the device and many other things. So we're planning on using that activities to be sent to, for example, for remote monitoring applications when the decision is made on the wearable device. For care plans and maybe family just watching them to see if they're taking care of themselves and things like that.
Okay, so just looking at healthy older adults as opposed to maybe those that have fallen and or had some kind of injury and maybe having some kind of rehabilitation.
Yeah, it could be an application as well, but in general, because we're just looking at most of them don't find them comfortable to wear. So it was just an application to see, if we use this technique to where the user wakes up today and say, I don't want to wear a device, I'll just put it in my pocket, and we can still get meaningful data to monitor them in that sense.
Right, I understand. And Sam had another question. Said she was really interested in your introduction, but wanted to ask the question: why isn't there good uptake or acceptance at the moment for these kind of wearables? And she wasn't sure if you'd mentioned how your work would increase the acceptance from a user perspective.
Right. So there's a lot more aspects. So this is just one approach that we're using to solve an adoption issue. So we did a co-design workshop where people we were talking with other others to see what we can do to make it more appealing to them. So they were saying, they find it sweaty to wear or things like that. So if they, for example, they want to wear their own jewelry and things like that. So if they don't want to wear it on their wrist, they can place it on their pockets. So we can still get that meaningful data out of it. So they wanted to integrate into their daily life as it is, not to change anything. So in terms of adoption by itself, I feel like mostly it's just the user interface, and also the issue of like most of them feel like there's much more energy required to learn to use these devices. So I feel like the other approaches that we're taking are more so like to reduce the learning curve as little as possible, to make it so like even the people that have no technology experience can get to use these technologies as well.
Right. Yeah, very good point. Roxana has asked, she just got one question. What is influencing the accuracy? I don't know if, Roxana, you need to expand on that at all. It might take her a second. Do you understand what she's asking for there? So what are the influences
Not really,
what are the influences, what are the external factors, if you like, that are affecting the accuracy of your data?
I think it could be just there's higher accuracies for the locations where there's less movement, I think. So I think it's more so to do with variations, and we've seen a huge difference with using the two types of sensors. The main restriction that we've had is also most of the devices on the market, you can't actually manipulate or change anything. So the device that we're using is an open source device. So it only comes with an accelerometer and a magnetometer. So there's very compared to like an IME with a gyroscope, it would get more out of that than with the two sensors that we have. So with this one, it was only accelerometer data, but we've been able to use the accelerometer and the magnetometer in the new 10 participant data that we have, and we've seen much more improvement in classifying the different activities. So I think it's more so to do with orientation on how we place the wearable device in the pockets or in the when you're placing it in the direction of the wrist. So I feel like those are affecting how accurate the data we're getting. I think the differences in affecting the accuracy of the allocations, I think.
It would be quite interesting to look at using a more accurate device and doing a repeatable experiment to compare what the sort of market technology compared to sort of very sensitive devices would be, to look at what the difference would be, perhaps.
Yeah, that's what we were planning as well, because we've been able to identify some other wearable devices that are open source as well that we can able to implement apart from the Bango.js too. And also another thing was with the other models that we're looking into comparing with this, because this was a very basic model with no actual contribution in how the model is designed and everything. And there's been a lot of research on human activity recognition. So we're trying to see, because most of them are very they don't address their position differences. So we're looking to see if we can use these models to then apply it to this particular case to see if the different models will then affect how everything works.
Yeah. Sam's also, or Samantha's asked another question. She said she'd be very interested to see progress beyond the 4 activities that you've focused on. And indeed, Alexandra pointed out you picked quite a difficult one of going sort of ascending and descending. Yeah. What are your thoughts on how you might improve on the work that you've done now and how you might take that forward?
I think I did my the formulation of how to address this prematurely, because we did the co-design workshop and they were just like, they would prefer to wait on different places, but I didn't specifically ask what they would. I feel like it would have been better to go hand in hand in if they were to be in a situation where they're being monitored, what activities are important to them, and see how we can implement that into this. So I feel like that would be an important aspect. But we do have another co-design workshop to get more insight on how we can make this better for them.
I think that's a very good point in terms of whenever you're doing work in this kind of environment, you have to do those co-design workshops. You have to be working directly with patients and take it beyond the laboratory, as it were, into the real world. So thank you. Yeah. Has anybody else got any questions before? No, we're all good. Well, thank you very much for coming and giving your presentation. It was really insightful. Thank you for taking part in this. And yeah.
Hello, everyone. So I'm Yanni, and I'm here to talk about producing microfinite element models from real-time clinical CT scanners, calibration, validation, and material mapping strategies. So we're talking about CT scanners because CT scans are very important. They're useful to acquire precise geometry of our body inside our skin without the need to carry it out and see with our own eyes. And it's also allowing us to see patient-specific geometry with anatomical accuracy. And nowadays, we're also able to do simulations with FEA models created from CT scans that are improving diagnosis and pre-op planning and stuff like that. So right now, there are 3 main types of CT scanners. So the mostly used one is the conventional one. So the patient is laying down on a table and is used clinically. So the radiation is okay. Is a normal radiation, normal 1-day radiation per person on planet Earth, and the resolution is quite okay for the majority of stuff. Then there's the micro CT scanner, which is using my in vitro research because the radiation is very, very high. So it's about 100 times the conventional radiation, but the resolution is also very, very much higher. So it can only be used on small animals or animated objects or bone samples and stuff like this. So nowadays there's also a new technology called the standing CT scanners that basically the patient is able to stand instead of lying down on the table, and the radiation is way lower than the micro or even the conventional one, but the resolution is better than the conventional, even though not as good as the micro CT scanners. But the problem of these scanners is that they haven't been properly validated for the simulation part, so we're here to do this. So we use basically 5-chip vertebrae to do this validation and we put them in a compression test. So we acquired some data from the compression test. We also did some machine compliance, studied the machine compliance, and we found out this equation that could allow us to figure out how much the machine itself was compressing, and we could find the real data of the samples. And for the simulation part, we scanned these 5-chip vertebrae in a scanner, which is a standing CT scanner, and we also used two phantoms. They were checking the acquired image was fine, and we also made a mesh convergence study that was making sure that the simulation mesh was not too fine or too coarse and was giving us the right results with the right amount of time. So with also with the CT scans, now we know that there's a correlation between the grayscale values and the material properties. So for this reason, we have used from literature for equations that basically mapped from the CT scans to the simulation models that with material properties that were realistic to the ones of the bone sample. And we have used these three, and then another one with another software called Metal with these parameters. And then we use rigid body constraints as boundary conditions. And then we apply the displacement, which is approximately 0.8% of the height of the sample, because the yield point of bone is about 1%. We didn't want to go anywhere near the plastic behavior of bone. And then the sample was fixed at the bottom. This is an example of the results that we got for each sample. So you can see that the blue dots are the results, from the experimental data. We can see that there's an initial shoulder where basically the pad, the upper and lower pads were adhering to the bone samples, and so the experimental data was not linear yet. And then in the red line is the best fit line of the linear region of the bone sample, and the yellow, the blue, the gray, and the green lines are the results from the simulation we did with the 4 different equations. We could see that they give us very different results. So this is a summary of the 5 bone samples, and we can see that overall equation A and equation C, which are the gray and the blue and the light blue lines, are the equations that behave the best, they estimated the best the stiffness of the bone samples, while equation B, yellow, overestimated in 4 cases, and then was accurate in just one, while metal, the green line, underestimated in 4 vertebrae and overestimated in the last one. So as you can see, we used different types of boundary conditions to see if that was going to change the results in any way, and because we decided to use rigid body constraints, but obviously we could have used pads and directly, but there were some interference problem between the bone and the pad, so we had to do some changes to avoid mistakes. And we've seen though that the difference in results were very, very minimal. So at the end, we didn't really consider them, and we just used the rigid body constraints. So, and we made this validation study because we wanted to improve the diagnosis and the situation for the flat foot cases, because flat foot is a condition that many people have on the in our population, and it can be as extreme that it requires surgery, but the outcomes are variable because it is condition that it can be different according to the person, and the influencing factors are completely not understood, so we don't really know what it causes it and why it causes it and stuff like this. So that's why we want to improve surgery outcomes, and we want to know how surgery can be different on different patients. For this reason, we did this study because standing CT scanners can be very important for flat foot people, because flexible flat foot is visible on weight bearing, which means that it's visible when the patient is standing up, but not when the patient is lying down on a table, because it looks like a normal foot. So in this case, with flat foot case, standing CT scans are very, very important. So for this reason, we made this study. So, and thank you for listening.
Thanks, Yanni. That was really interesting. Thank you for your lovely presentation. I have a question if no one else wants to jump in. That's all right.
Yeah, absolutely. Go ahead.
I've got a few questions. I'm going to ask the really stupid question first and get it out of the way. That's all right. So really interesting to see how you're planning on using that work. Why can't I just look at someone's foot and see whether or not they have a flat foot?
Yes, because the thing is, there are so many different. Obviously, flat foot is not something that you just say, okay, this patient doesn't have any arch, so it's flat. It's not that immediate, if you know what I mean. So it's like, even though, for example, right now, so in surgery, they make surgery just for people that have pain in their flat foot. So, for example, if one patient comes in and has the flattest foot ever, but it doesn't have any pain associated with it, like running or doing any sports, for example, the NHS doesn't do any surgery on it because it doesn't cause you any pain. But for example, another foot that maybe can be a little bit flat, but not too much. But for example, it stops you from doing some activity, or maybe it's uncomfortable for you to walk or to run or to do any other thing. For example, they actually make surgery on it. So as I said, is a very kind of is not so immediate to understand why we have pain and why we need surgery. So that's why we need to understand what's happening in the with like the how the bones are related to each other and what's causing the pain, because actually it's not like with the I don't know, with the knee or maybe the elbow, that you just have two or three bones connected to each other and it's just immediate because it's just two, three bones. But in the case of the foot, we're talking about, I don't know, 20, 30 bones connected to each other, and you don't really know where the pain is coming from and why. So
Okay.
that's why you need to, go underneath the skin and understand what's happening.
And so I guess following up from that, then what would be your next steps? This is your PhD research, right?
Yes.
Am I right?
Yes.
And where are you in your PhD at the moment?
So at the moment, I'm in my writing up stage. So I'm at the
Fab.
end. So, but
Congratulations.
Thank you. I'm not done yet, but I'm very close to finishing. And so my PhD had some difficulties. I should have done a way bigger project than I had, because my PhD I had to study through COVID and everything, and obviously the hospitals were not really talking to me when there was the pandemic. But right now my study is more about, for example, I validated this and I tried to understand, the shape of the bones, in that if that is affecting the flat foot in any way or not, because obviously as you may know, the shape of our bones, it kind of depends on how we live and how we use our body. So we want to understand if, for example, if a flat foot patient, for example, has the same bone shape as a normal foot person, and we want to understand if there are any changes in that. Because, for example, when people actually perform surgery on a flat foot, they change the shape of the bones to make sure that they create an arch in the foot. And we want to understand, for example, if the flat-foot people have bones with a different shape, that could, change the outcome of the surgery, because maybe the surgeon doesn't really know that their bone is somewhat different, and maybe the outcomes may be bad or may be good. So we want to understand flat-foot in general. So it's such I mean, really, there are so many ways to to improve these kind of topic. In origin, I wanted to do an FEA, a simulation of the movement of the whole foot, to understand where the pain was coming from in flat foot patient. But at the end of the day, I couldn't do that study, but that could have been very, very interesting, for example.
Thank you.
Can I ask a question, if that's all right, Yanni? Thank you.
Yeah,
It's
sure.
a really interesting presentation. Did you say that it was sheep vertebrae that you were using for your initial
Yes.
characterization? So how do you think then that the sheep vertebrae are going to differ from the bones of the foot that you're wanting to characterize, and obviously sheep to human population? How do you translate that in your models?
So we have seen that through the literature that other cases have demonstrated that sheep bones are very, very similar to human bones. So there's a correlation between them, and it's kind of a proven case that the sheep bones can be used instead of human bones. So I guess that since the physics is the same, I guess that scanning sheep bones and scanning human bones is kind of the same result
Do you
because of
think they might,
literature.
with the sheep, obviously with their spines, they're not necessarily loading on like the foot might load?
Yes,
Do you
but
think the soft tissues and anything around that environment on top of your bones, when you're talking about wanting to do that full foot model, do you think having an understanding around that loading condition might influence how you change the loading between obviously sheep vertebra and your foot at all?
uh, I mean, it may be a little bit different, but I think that maybe the differences can be, are not that big, that have to be considered, I think, because after all, we're talking about cancellous bone either way. So even in the foot bones, we're not talking about long bones or very strong bones like the femur. We're talking about very small bones. So we're talking about mostly cancellous bone. So after all, quite similar to sheep vertebra in general. And I think that the objective of this study was to understand if the standing CT scanners could scan uh, a bone sample, whichever a bone sample, and the simulation could be faithful to the experimental setting. So we kind of demonstrated that with this study. So I don't think that even with soft tissues and other soft tissues or ligaments and stuff like that, I don't think that the results would change in any way the results, if you know what I mean.
Thank you.
Yanni, I have a question from Roxana, who's online but not able to dial in with a video. And I think you've answered part of her question already in terms of what motivated you to focus on this particular group of bones. But I want to go on to a second question, which was building on the findings that you've generated so far. What other unmet needs do you think that this methodology might potentially address?
Methodology, I don't know about methodology, but I think that the results of this study can be used in many, many different ways, because flatfoot is just one condition that can be influenced by standing to discount. Because, for example, we have seen that even lungs can change their shape and their volume according to if the patient is lying down or standing up, and even other kind of conditions, if not related to lungs or other things, can be affected by the fact if the patient was scanned while lying down or not. So the results of this study can be used in very, very different cases that are not related to flatfoot in any way. Flatfoot is just the reason why I did this study, but I think that the standing CT scans can be useful for many other reasons that I wrote and published an article about this study. So I've seen that there are many other things that are, there can be, you know, other reasons that standing CT scans may be useful for them, and these, you know, this validation between scans and actual simulation can be useful for those cases to study better those cases that are not related to bones or flatfoot in any way.
Okay. And a question from me, actually. So you mentioned right at the beginning that the technology, the CT scanners, the sitting CT scanners have yet to be validated in terms of their data. How do you think that the work that you've done here could help the manufacturer of that device to validate their technology?
I think maybe I wasn't clear enough. I mean, I didn't say validated to be used in clinical setting because they are already used in clinical setting, maybe not very, very frequently, but they are already being used in clinical setting, but they haven't been validated, you know, with simulation setting. So we weren't sure that actually the shape of the scanned object was going to be faithful to the shape of the simulation object, if you know what I mean. So we have validated this just so the images for simulation only, but they already have been used in clinical settings already.
Right, I understand. Were you able to compare the difference between the standing scans and the conventional scans at all? Did you compare any of that data while you were doing the work?
As you can see, I mean, not really, but but you can see it from yourself from these slides, because you see at the bottom that they are vertebrae, so these are conventional micro standing, and you can see that the conventional you can't see any trabeculae at all. But in micro, obviously, you can see them very, very clearly, and the standing, you can kind of see them. And I think that the work I've been doing in this study, which I converted the material properties from the grayscale of the CT scans, was possible with standing CT scans, but not with conventional, because as you can see, it's just a solid gray, and there's no much difference between one pixel with another. So I don't think that I could have done the same kind of study using conventional CT scans.
Thank you. Would anybody else like to ask any further questions? No? Okay. Well, Yanni, thank you very much for that presentation. Really insightful.
Thank you.
And thank you for taking the time.